Ribosome. Ribosomes, /ribosome, named also Palade's corpuscles, comprise a complex macromolecular machine, found within all living cells, that serves as the site of biological protein synthesis, translation. Ribosomes link amino acids together in the order specified by messenger RNA, mRNA, molecules. Ribosomes consist of two major components, the small ribosomal subunits, which read the mRNA, and the large subunits, which join amino acids to form a polypeptide chain. Each subunit consists of one or more ribosomal rRNA molecules and a variety of ribosomal proteins, rProtein or rProtein. The ribosomes and associated molecules are also known as the translational apparatus. Overview. The sequence of DNA, which encodes the sequence of the amino acids in a protein, is copied into a messenger RNA chain. It may be copied many times into RNA chains. Ribosomes can bind to a messenger RNA chain and use its sequence for determining the correct sequence of amino acids for generating a given protein. Amino acids are selected and collected and carried to the ribosome by transfer RNA, rRNA, molecules, which enter one part of the ribosome and bind to the messenger RNA chain. It is during this binding that the correct translation of nucleic acid sequence to amino acid sequence occurs. For each coding triplet in the messenger RNA there is a distinct transfer RNA that matches and which carries the correct amino acid for the coding triplet. The attached amino acids are then linked together by another part of the ribosome. Once the protein is produced, it can then fold to produce a specific functional three-dimensional structure, although during synthesis some proteins start folding into their correct form. A ribosome is made from complexes of RNAs and proteins and is therefore a ribonucleoprotein. Each ribosome is divided into two subunits. When a ribosome finishes reading an mRNA molecule, these two subunits split apart. Ribosomes are ribozymes, because the catalytic peptidyl transferase activity that links amino acids together is performed by the ribosomal RNA. Ribosomes are often associated with the intracellular membranes that make up the rough endoplasmic reticulum. Ribosomes from bacteria, archaea and eukaryotes in the three-domain system resemble each other to a remarkable degree, evidence of a common origin. They differ in their size, sequence, structure, and the ratio of protein to RNA. The differences in structure allow some antibiotics to kill bacteria by inhibiting their ribosomes, while leaving human ribosomes unaffected. In bacteria and archaea, more than one ribosome may move along a single mRNA chain at one time, each reading its sequence and producing a corresponding protein molecule. The mitochondrial ribosomes of eukaryotic cells are produced from mitochondrial genes, and functionally resemble many features of those in bacteria, reflecting the likely evolutionary origin of mitochondria. Discovery. Ribosomes were first observed in the mid-1950s by Romanian-American cell biologist George Emil Palade, using an electron microscope, as dense particles or granules. The term ribosome was proposed by scientist Richard B. Roberts in the end of 1950s. Albert Claude, Christian Nadeau, and George Emil Palade were jointly awarded the Nobel Prize in Physiology or Medicine, in 1974, for the discovery of the ribosome. The Nobel Prize in Chemistry 2009 was awarded to Venkatraman Ramakrishnan, Thomas A. Steitz and Ada E. Yanath for determining the detailed structure and mechanism of the ribosome. Structure. The ribosome is a highly complex cellular machine. It is largely made up of specialized RNA known as ribosomal RNA, rRNA, as well as dozens of distinct proteins. The exact number varies slightly between species. The ribosomal proteins and rRNAs are arranged into two distinct ribosomal pieces of different size, known generally as the large and small subunit of the ribosome. Ribosomes consist of two subunits that fit together, Figure 2, and work as one to translate the mRNA into a polypeptide chain during protein synthesis, Figure 1. Because they are formed from two subunits of non-equal size, they are slightly longer in the axis than in diameter. Bacterial ribosomes. Prokaryotic ribosomes are around 20 nanometers, 200 A ring, in diameter and are composed of 65% rRNA and 35% ribosomal proteins. Eukaryotic ribosomes are between 25 and 30 nanometers, 250 to 300 A ring, in diameter with an RNA to protein ratio that is close to 1. Crystallographic work has shown that there are no ribosomal proteins close to the reaction site for polypeptide synthesis. This suggests that the protein components of ribosomes do not directly participate in peptide bond formation catalysis, but rather that these proteins act as a scaffold that may enhance the ability of RNA to synthesize protein, c. ribosome. The ribosomal subunits of bacteria and eukaryotes are quite similar. The unit of measurement used to describe the ribosomal subunits and the RNA fragments is the Spedberg unit, a measure of the rate of sedimentation and centrifugation rather than size. This accounts for why fragment names do not add up. For example, bacterial 70S ribosomes are made of 50S and 30S subunits. Bacteria have 70S ribosomes, each consisting of a small, 30S, and a large, 50S, subunit. E. coli, for example, has a 16S rRNA subunit, consisting of 1,540 nucleotides, that is bound to 21 proteins. The large subunit is composed of a 5S rRNA subunit, 120 nucleotides, a 23S rRNA subunit, 2,900 nucleotides, and 31 proteins. Affinity label for the Rna binding sites on the E. coli ribosome allowed the identification of A and P site proteins most likely associated with the peptidyl transferase activity. Labeled proteins are L27, L14, L15, L16, L2. At least L27 is located at the donor site, as shown by E. Kalatz and A. P. Zernilovsky. Additional research has demonstrated that the S1 and S21 proteins, in association with the 3' of 16S ribosomal RNA, are involved in the initiation of translation. Eukaryotic ribosomes. Eukaryotes have 80S ribosomes located in their cytosol, each consisting of a small, 40S, and large, 60S, subunit. Their 40S subunit has an 18S RNA, 1900 nucleotides, and 33 proteins. The large subunit is composed of a 5S RNA, 120 nucleotides, 28S RNA, 4700 nucleotides, a 5.8S RNA, 160 nucleotides, subunits and 46 proteins. During 1977, Zernilovsky published research that used affinity labeling to identify Rna binding sites on rat liver ribosomes. Several proteins, including L32/33, L36, L21, L23, L28/29 and L13 were implicated as being at or near the peptidyl transferase center. Plastiribosomes and mitoribosomes. In eukaryotes, ribosomes are present in mitochondria, sometimes called mitoribosomes, and in plastids such as chloroplasts, also called plastiribosomes. They also consist of large and small subunits bound together with proteins into one 70S particle. These ribosomes are similar to those of bacteria, and these organelles are thought to have originated as symbiotic bacteria of the two. Chloroplastic ribosomes are closer to bacterial ones than mitochondrial ones are. Many pieces of ribosomal RNA in the mitochondria are shortened, and in the case of 5S RNA, replaced by other structures in animals and fungi. In particular, Leishmania tarantulae has a minimalized set of mitochondrial RNA. The cryptomonad in chlorarachnophyte algae may contain a nucleomorph that resembles a vestigial eukaryotic nucleus. Eukaryotic ADS ribosomes may be present in the compartment containing the nucleomorph. Making use of the differences. The differences between the bacterial and eukaryotic ribosomes are exploited by pharmaceutical chemists to create antibiotics that can destroy bacterial infection without harming the cells of the infected person. Due to the differences in their structures, the bacterial 70S ribosomes are vulnerable to these antibiotics while the eukaryotic ADS ribosomes are not. Even though mitochondria possess ribosomes similar to the bacterial ones, mitochondria are not affected by these antibiotics because they are surrounded by a double membrane that does not easily admit these antibiotics into the organelle. The same cannot be said of chloroplasts, where antibiotic resistance in ribosomal proteins is a trait to be introduced as a marker in genetic engineering. Common properties. The various ribosomes share a core structure, which is quite similar despite the large differences in size. Much of the RNA is highly organized into various tertiary structural motifs, for example pseudoknots that exhibit coaxial stacking. The extra RNA in the larger ribosomes is in several long continuous insertions, such that they form loops out of the core structure without disrupting or changing it. All of the catalytic activity of the ribosome is carried out by the RNA. The proteins reside on the surface and seem to stabilize the structure. High-resolution structure. The general molecular structure of the ribosome has been known since the early 1970s. In the early 2000s, the structure has been achieved at high resolutions, of the order of a few angstroms. The first papers giving the structure of the ribosome at atomic resolution were published almost simultaneously in late 2000. The 50S, large prokaryotic, subunit was determined from the archaean Haloarcula marismortui and the bacterium Deinococcus radiodurans, and the structure of the 30S subunit was determined from Thermus thermophilus. These structural studies were awarded the Nobel Prize in Chemistry in 2009. In May 2001, these coordinates were used to reconstruct the entire T Thermophilus 70S particle at 5.5A ring resolution. Two papers were published in November 2005 with structures of the Escherichia coli 70S ribosome. The structures of the vacant ribosome were determined at 3.5A ring resolution using X-ray crystallography. Then, 2 weeks later, a structure based on cryo-electron microscopy was published, which depicts the ribosome at 11 to 15A ring resolution in the act of passing a newly synthesized protein strand into the protein-conducting channel. The first atomic structures of the ribosome complex with rRNA and mRNA molecules were solved by using X-ray crystallography by two groups independently, at 2.8A ring and at 3.7A ring. These structures allow one to see the details of interactions of the Thermus thermophilus ribosome with mRNA and with rNAs bound at classical ribosomal sites. Interactions of the ribosome with long rNAs containing Schindel-Garneau sequences were visualized soon after that at 4.5 to 5.5A ring resolution. In 2011, the first complete atomic structure of the eukaryotic ADS ribosome from the yeast Saccharomyces cerevisiae was obtained by crystallography. The model reveals the architecture of eukaryote-specific elements and their interaction with the universally conserved core. At the same time, the complete model of a eukaryotic 40S ribosomal structure in Tetrahymena thermophila was published and described the structure of the 40S subunit, as well as much about the 40S subunit's interaction with IF1 during translation initiation. Similarly, the eukaryotic 60S subunit structure was also determined from Tetrahymena thermophila in complex with IF6. Function. Ribosomes are minute particles consisting of RNA and associated proteins that function to synthesize proteins. Proteins are needed for many cellular functions such as repairing damage or directing chemical processes. Ribosomes can be found floating within the cytoplasm or attached to the endoplasmic reticulum. Basically, their main function is to convert genetic code into an amino acid sequence and to build protein polymers from amino acid monomers. Ribosomes act as catalysts in two extremely important biological processes called peptidal transfer and peptidal hydrolysis. The PT center is responsible for producing protein bonds during protein elongation. Translation. Ribosomes are the workplaces of protein biosynthesis, the process of translating mRNA into protein. The mRNA comprises a series of codons which are decoded by the ribosome so as to make the protein. Using the mRNA as a template, the ribosome traverses each codon, 3 nucleotides, of the mRNA, pairing it with the appropriate amino acid provided by an aminoacyl rNA. Aminoacyl rNA contains a complementary anticodon on one end and the appropriate amino acid on the other. For fast and accurate recognition of the appropriate rNA, the ribosome utilizes large conformational changes, conformational proofreading. The small ribosomal subunit, typically bound to an aminoacyl rNA containing the first amino acid methionine, binds to an August codon on the mRNA and recruits the large ribosomal subunit. The ribosome contains 3 RNA binding sites, designated A, P and E. The A site binds an aminoacyl rNA or termination release factors, the P site binds a peptidal rNA, a rNA bound to the polypeptide chain, and the E site, exit, binds a free rNA. Protein synthesis begins at a start codon August near the 5' end of the mRNA. mRNA binds to the P site of the ribosome first. The ribosome recognizes the start codon by using the Schindel-Garneau sequence of the mRNA in prokaryotes and Kozak box in eukaryotes. Although catalysis of the peptide bond involves the C2-hydroxyl of RNA's P site adenosine in a proton shuttle mechanism, other steps in protein synthesis, such as translocation, are caused by changes in protein conformations. Since their catalytic core is made of RNA, ribosomes are classified as ribozymes, and it is thought that they might be remnants of the RNA world. In Figure 5, both ribosomal subunits, small and large, assemble at the start codon, towards the 5' end of the mRNA. The ribosome uses rRNA that matches the current codon, triplet, on the mRNA to append an amino acid to the polypeptide chain. This is done for each triplet on the mRNA, while the ribosome moves towards the 3' end of the mRNA. Usually in bacterial cells, several ribosomes are working parallel on a single mRNA, forming what is called a polyribosome or a polysome. Co-translational folding. The ribosome is known to actively participate in the protein folding. The structures obtained in this way are usually identical to the ones obtained during protein chemical refolding. However, the pathways leading to the final product may be different. In some cases, the ribosome is crucial in obtaining the functional protein form. For example, one of the possible mechanisms of folding of the deeply knotted proteins relies on the ribosome pushing the chain through the attached loop. Addition of translation-independent amino acids. Presence of the ribosome quality control protein RQC2 is associated with mRNA-independent protein elongation. This elongation is a result of ribosomal addition, via rNAs brought by RQC2, of cattails. Ribosomes extend the C-terminus of a stalled protein with random, translation-independent sequences of alanines and threonines. Ribosome locations. Ribosomes are classified as being either free or membrane-bound. Free and membrane-bound ribosomes differ only in their spatial distribution, they are identical in structure. Whether the ribosome exists in a free or membrane-bound state depends on the presence of an targeting signal sequence on the protein being synthesized, so an individual ribosome might be membrane-bound when it is making one protein, but free in the cytosol when it makes another protein. Ribosomes are sometimes referred to as organelles, but the use of the term organelle is often restricted to describing subcellular components that include a phospholipid membrane, which ribosomes, being entirely particulate, do not. For this reason, ribosomes may sometimes be described as non-membranous organelles. Free ribosomes. Free ribosomes can move about anywhere in the cytosol, but are excluded from the cell nucleus and other organelles. Proteins that are formed from free ribosomes are released into the cytosol and used within the cell. Since the cytosol contains high concentrations of glutathione and is, therefore, a reducing environment, proteins containing disulfide bonds, which are formed from oxidized cysteine residues, cannot be produced within it. Membrane-bound ribosomes. When a ribosome begins to synthesize proteins that are needed in some organelles, the ribosome making this protein can become membrane-bound. In eukaryotic cells this happens in a region of the endoplasmic reticulum, called the rough ER. The newly produced polypeptide chains are inserted directly into the ER by the ribosome undertaking vectorial synthesis and are then transported to their destinations, through the secretory pathway. Bound ribosomes usually produce proteins that are used within the plasma membrane or are expelled from the cell via exocytosis. Biogenesis. In bacterial cells, ribosomes are synthesized in the cytoplasm through the transcription of multiple ribosome gene operons. In eukaryotes, the process takes place both in the cell cytoplasm and in the nucleolus, which is a region within the cell nucleus. The assembly process involves the coordinated function of over 200 proteins in the synthesis and processing of the rNAs, as well as assembly of those rNAs with the ribosomal proteins. Origin. The ribosome may have first originated in an RNA world, appearing as a self-replicating complex that only later evolved the ability to synthesize proteins when amino acids began to appear. Studies suggest that ancient ribosomes constructed solely of RNA could have developed the ability to synthesize peptide bonds. In addition, evidence strongly points to ancient ribosomes as self-replicating complexes, where the RNA in the ribosomes had informational, structural, and catalytic purposes because it could have coded for rNAs and proteins needed for ribosomal self-replication. Hypothetical cellular organisms with self-replicating RNA but without DNA are called ribocytes, or ribocells. As amino acids gradually appeared in the RNA world under prebiotic conditions, their interactions with catalytic RNA would increase both the range and efficiency of function of catalytic RNA molecules. Thus, the driving force for the evolution of the ribosome from an ancient self-replicating machine into its current form as a translational machine may have been the selective pressure to incorporate proteins into the ribosome's self-replicating mechanisms, so as to increase its capacity for self-replication. Specialized ribosome. Many textbooks suggest that there are only two kinds of ribosomes, namely prokaryotic and eukaryotic ribosomes. However, ribosomes are surprisingly heterogeneous with different compositions in different species. Heterogeneous ribosomes have different structures and thus different activities compared to typical ribosomes in major model organisms. Heterogeneity in ribosome composition has been proposed to be involved in translational control of protein synthesis. Vincent Morrow and Gerald Edelman proposed the ribosome filter hypothesis to explain the regulatory functions of ribosomes. Emerging evidence has shown that specialized ribosomes specific to different cell populations can affect how genes are translated. Some ribosomal proteins exchange from the assembled complex with cytosolic copies, suggesting that the structure of the in vivo ribosome can be modified without synthesizing an entire new ribosome. Ribosomal proteins. A group of highly acidic ribosomal proteins, RPs, also known as P proteins, are known to be present on the 60S subunit in multiple copies in the ribosome stalk, and P proteins mediate selective translation. These P proteins can be found in yeast and mammalian cells. If P proteins are not present in yeast, this can cause the yeast to have a cold-sensitive phenotype. If P proteins are not present in human cells, this could cause autophagy induction. Certain ribosomal proteins are absolutely critical while others are not. For instance, RPL28 and RPL5 mutant flies are alike but have abnormally large wings. RPL38 also appears to be critical in mammals only under very specific conditions. In mice, RPL38 is required for the translation of a subset of Hox's mRNA, and a mutation of RPL38 lead to a homeotic transformation with a short tail. Other ways heterogeneity can arise from post-translational modifications to RPs include acetylation and methylation in species like yeast, Arabidopsis, and human cells. But there is no overall change in protein synthesis. Modifications to core ribosomal proteins, RPs, can also give rise to the formation of heterogeneous ribosomes. For instance, in yeast, RPL28 ubiquitination levels vary with the cell cycle. Ribosomes with the polyubiquitinated RPL28 carry out protein synthesis at a higher rate in vitro compared with ribosomes with monubiquitinated RPL28. Ribosome-associated factors. Heterogeneous ribosomes can also arise from other factors binding to the ribosome surface. For example, ribosome-associated factor, RAC1, is so tightly associated with the ribosome that its binding is resistant to high salt washes in vitro. RAC1 is required for efficient translation of mRNA with short open reading frames. RNA heterogeneity. Viral layers, internal ribosome entry site, translations can also be mediated by specialized ribosomes. Specifically, 40S ribosomal units without RPS25 in yeast and mammalian cells are unable to begin translation from two viral irises, namely the hepatitis C virus IRS and the cricket paralysis virus, CRPV, intergenic region. The CRPV only needs RPS25 to begin translation and mediate ribosome recruitment. Usually, if RPS25 is not present in a certain IRS, initiation from these irises can be defective. RNA modifications. Heterogeneity of ribosomal RNA modifications plays an important role in structural maintenance and/or function, such as modulating translation, and most mRNA modifications are found in highly conserved regions. The most common RNA modifications are pseudorytylation and 2-O-methylation of ribose, or pseudorytylation and 2-O-methylation of ribose, or pseudorytylation and 2-O-methylation of ribose, or pseudorytylation